Thank you. I'm told I need no introduction, so you're not going to get one. Right, we're going to talk about epidemics, pandemics, and surveillance today. And I would like to see this pointer behave itself. Can we see a pointer? Right. Pointer option, laser pointer. There we go. So, to give you some background and to make it clear what exactly people are looking for, I've already told you something about how you name influenza viruses. Well, they're named according to this rather impossible-looking scheme. That's serotype, host species, site of origin, strain, year, isolated, and HA and NA type, which comes up with something like this. This was actually isolated, this virus, A. Hearn, South Africa, strain 1, 1961, H5N2. It was the first time that people had found H5, which you might remember from H5N1, from a seabird. This was done at UCT in the so-called Virus Research Unit by a guy called Wally Becker. Then A. New Caledonia, 1999. This is the 20th isolate of virus from New Caledonia. It's a subtype A, and it's H1N1. So when you see a virus name that looks something like that, you have an idea that it tells you both what kind of virus it is, influenza A or B, where it came from, when it came from, and the exact H and N values. So a B isolate, for example, because there's only 2 variants of these, was just called B Malaysia 2004. So to remind you, pandemic viruses that we have managed to type accurately, H2N2 up to about 1900, and note from about 1890 something onwards. H3N2 pops up around about 1900, last till 2018. 1918. H1N1 takes over till 1957. You have an H2N2 pandemic that only lasts 10 years before it's replaced by H3N2, another pandemic, which has lasted till the present day. In fact, right now in Brisbane, Australia, they're having a late or unseasonal outbreak of H3N2. Then 1977, H1N1 pops up again. And the interesting thing was it was more or less identical to the virus that ended in around about 1957, meaning that it had been sitting in a freezer somewhere. And the speculation is rife, and if you get my book, which I'm shamelessly advertising on the ends of the benches, you can read the purported history. That one lasted until 2009, when another H1N1 popped up. And I'm going to explain some of this, how these happen. Because remember, pandemics, lots of people die, huge incidence of disease because you're not immune to this new virus that pops up. They're followed by seasonal circulation of the same virus, which replaces the previous seasonal strain or strains. Now, recent years are pretty unusual in that we've had H3N2 and H1N1 of one or other type circulating since 1968 for 50 years. That has not happened before with subtype A viruses. Now, this was an Economist cover from some years ago advertising a book called Pale Rider. And I emphasize again, up to 100 million people died. The conservative estimate is 50. The real estimate is probably 100 because there are so many unaccounted cases. And there's something right from here, near at home. Brian Davey in Cape Town subscribes to a Facebook site called Cape Town Down Memory Lane. He had this headstone from the Plumstead Cemetery. And I immediately said, please, 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 give it to me. And he very kindly did. This memorializes Susannah, Beatrice, or Sunshine Hearns. Note her age of birth. She died in 1918, but she was born in 1885, which puts her in exactly the right age group for the maximum mortality of that virus, around about 35 odd. That was the age group that got hit the hardest. She was a staff nurse working with the military in 1918, and as I said, died doing her duty. So how do we get there? How do we pop up with the pandemic? So a little bit of reiteration here. It's generally a mixture between human viruses and, for example, bird viruses. That can happen inside a single cell. It's not unknown for an organism to get infected with more than one virus at the same time. If that happens, individual cells can be infected with two different disease agents. And if that happens with flu, the components of those viruses can mix together and pop out something new. So, in terms of pandemics and disease, pre-1957, I was born before 1957, so I was almost certainly infected with the circulating H1N1 back then. And then probably again, this was the H1N1 that was circulating from 1918. It was now mild disease. The virus had in fact adapted, but also the population was largely immune. 1957, a brand new virus popped up, H2N2. Two new proteins on the outside, a completely different coat. If you look at that compared to that, everything on the outside is different. Very few of the population were immune. The few that were immune, because there was an H2N2 up to about 1900, were elderly and therefore not particularly immune. So it infected a lot more people, caused severe disease, and over 3 million people died. And strangely enough, 1968, there was a replacement, H3N3. Take a look at what color remains the same. That's the neuraminidase protein here. The hemagglutinin completely changes. So people had partial immunity, caused moderately severe disease. It was not a nice virus, but people were partially immune. But over 1 million people died of that pandemic. And because we've forgotten, that was 50 years ago right now. As of early 1969, this virus reached its peak in the southern hemisphere, and that's from January onwards. So pandemic viruses don't care about seasons particularly. They circulate among the non-immune until people are immune or the virus dies out. Oh, and by the way, that was the first one that traveled predominantly at first by air, and that was US troops going home from guess where? Vietnam. So all that spread around there and back across that way and across that way to the US was air travel of troops. Then it started hitting the seaports and being transported to places like Africa and South America. 2009, we have a new pandemic, which is very mild, and people say, well, there was a huge amount of alarm over nothing. Not so quick. There's at least 250,000 people died of that virus, specifically that virus. Forget the other seasonal flus that were going around at the time as well. And it was reasonably mild because it was H1N1. And the interesting thing here is there is avian virus. These are 8 components of the virus genome, so 8 pieces of RNA. This is the virus that resulted down at the bottom here. That thing had contributions from a bird virus, an H1N1, the so-called classical swine flu that had been circulating since 1918. That's important to remember. Pigs got it from people in 1918. It circulated continuously in North America because this pandemic originated in Mexico. And in fact, it had broken out once before, 1976, so-called swine flu epidemic that caused a lot of alarm, and vaccines were made, and it petered out very quickly. The new version then got a dose of human flu, H3N2 components. So 3 different viruses mixing together in pigs comes out as a virus that is easily transmitted from pig to human and then from human to human, and causes reasonably severe disease, and strangely enough, especially among the same group of people who got hit by H1N1 in 1918. In other words, people, young adults. Fit young rugby players at Stellenbosch dropped dead of swine flu. Young athletes at UCT, people all over South Africa. It was disproportionately hit, that age group, again. Much less severe than the 1918 flu, but severe enough that people actually noticed. So, in layman's terms, this is what happened. Pig population in North America, you have the virus continuously circulating from 1918 to 2009. You then introduce components of a bird virus, and if you say how, well, it's an enteric virus in birds, literally transmitted in bird feces. Birds crap on pig pens. Pigs eat it. Literally, that's all you need. And then human virus. Pig handlers give pigs human viruses. Very well-known phenomenon. And the pigs returned the favor. They gave back that remixed virus in 2009 to pig handlers who took it home to their families, and it spread very, very rapidly indeed. But that's not the only one. There is a whole zoo of viruses. H5N1, that you've probably heard of, kills 50% at least of the people it infects. 50% case mortality rate. Luckily, it doesn't very easily at all transmit human to human. It's been circulating in livestock since 2003. H7N2, two cases in cats from the US. H9N2, this is bubbling under in China. It is increasingly appearing in people. Direct transmission from poultry. H7N7, H3N2 variant. Human virus got into pigs, is getting back into people at fairs, at livestock fairs in the US. H7N9, another matter for concern, down to H7N3. Here's another one, H5N6. It is circulating in Vietnam. Epizootic. In other words, it is circulating continuously in livestock and occasionally gets into people, and when you see it in people, it's severe. So that whole zoo has to be surveilled for. You want early warning if any of these things actually pops out into the human population and starts to spread. So, and getting on to what you really, really should do for yourself in March, April, the WHO recommendations for influenza vaccines for the southern hemisphere flu season right now, we don't get this vaccine, the quadrivalent vaccine, unless you really try really hard. And that would have two A types, the 2009 pandemic virus, an H3N2-like virus, and two B types. If, on the other hand, you get what is usually got by South Africa, the trivalent vaccine, that would be two A types, the pandemic 2009 and an H3N2, and one B type, the one that is most prevalent in the northern hemisphere in the last flu season. So we don't get the adjuvanted high-dose senior so-called vaccine. There's a way around this. You just get vaccinated twice. Don't say I said so. I'm not medic. You cannot take my advice. And I didn't say anything, and if you say I did, I'll say you lied. So how do you make eggs right now? I told you the other day that it's a 1942 technology. The first flu vaccines were made in 1942 by the US military. We still make them the same way. We use embryonated chicken eggs. So you take circulating flu virus, you mix it by that same technique I showed you, infecting the same cells with different viruses, with a strain of virus that you know multiplies well in eggs. You mix them until you get the coat proteins that you want, the HA and the NA of the circulating virus. These things then go through WHO labs. They are made at large scale, then partially purified, inactivated with something called beta-propiolactone or just formaldehyde, and the HA and NA proteins are split using a fractionation process. You get a so-called split vaccine, which still contains a whole lot of egg proteins, as a matter of interest, which is why people with egg allergies potentially at risk from egg-made vaccines. And one egg gives one dose of one virus. So to make a quadrivalent vaccine, you need 4 eggs, trivalent just 3. And if you think that if you want to vaccinate just a fraction of the Earth's population. That's a couple of 1,000 million eggs that you need in factories. So this gets scaled up to huge scale. We do not do this in Africa. You do it in Australia, they do it in Europe, and they do it in the US and some places in Asia. We don't do this. We don't have vaccine. So what do we need for pandemic vaccines? Well, problem is, it takes 6 months from identification of a virus. So if Ziad was still in that job and he surveilled human viruses and he found there's an H5N2 that does infect ostriches right now in South Africa, keeps on popping up, suddenly this thing's infecting ostrich handlers and it's being transmitted like wildfire. And that then goes to the WHO as an emergency and they say, we need a vaccine. Minimum 6-month period. It has not changed since the 1940s. We were too late with vaccine responses for 1957, 1968, and the 2009 pandemics. Those vaccines only came on stream when the pandemics were winding down. So we need more new and more effective technologies for much faster production. They do exist. It's just manufacturing inertia and the huge capital outlay involved that actually holds people back. And it's really necessary. This is a map of wild bird flyways. I show you this one here. This goes from far eastern Siberia all the way down to encompass Australia. Here's another one from Siberia through to India. Here's another one, Siberia all the way through to southern Africa. They are birds that migrate all the way through. The Hearn virus that I told you about just now was isolated in South Africa. An almost identically similar virus was isolated 6 months earlier in Scotland. Those birds had almost certainly, using flyways like this green one here, come down to the west coast of South Africa from birds from northern Europe. These viruses can fly as fast as birds do. Birds migrate from the far northern to the far southern hemisphere every year. And they're the ones who carry A-type viruses. These viruses could spread incredibly fast. So with that, let me leave you with a cartoon. This is one of the ones I liked from 2009. This, in the front boat, is the developed countries. That in the back boat is us, in the developing countries. There's another beautiful one of Pooh and Piglet with a very obscene caption that you could look up if you wanted to. Basically, if the pig sneezes, he's dead. That's where we were in 2010. We had no vaccine. The only vaccine in South Africa was for medical staff and for people working in the 2010 World Cup. So we really do have to start making our own, and the kind of surveillance that people like Ziad was doing lets us get there. Thanks. Shall I fire you up?
Great. Thank you, Ed. Okay, so I have been vaccinated, thanks. So during this week, we've learned how devastating the Spanish flu was. And today I'm going to be touching on how everything that we've learned in the last 100 years since the Spanish flu has impacted how we conduct surveillance and also how it affects our research efforts. Okay, so the big question is, why bother? Most people see flu as just the sniffles. We stay in bed, we get better, and we get back to work, right? It's just another respiratory disease. It's definitely not as attractive as many of the other viruses, like HIV or Ebola, or maybe saying attractive isn't the good word to use with HIV. But I'd like to put it to you that we actually feel that flu is more efficient at being a virus than most other viruses. As you know, a virus has no purpose. It is a blind machine, and that's a word that I took from Ed's lectures. Its only function is to replicate itself and spread. And flu does this amazingly well. It is capable of spreading across the world and keeping its victims alive long enough to have a greater geographic footprint. Okay, so why do we do flu surveillance? Right, this is the bottom line. What is the cost of it? So pneumonia is the primary cause of hospitalization of adults and children worldwide, and definitely in South Africa. And in South Africa, 9% of all hospitalized cases of pneumonia are caused by flu. So in our national surveillance programs for pneumonia, we found that around 19,500 flu-related hospitalizations occur every year. So if we try to put a number on that, which we have, we try to establish what the societal costs are. So this in the public sector would be the cost of treating individuals, the cost of money lost from days off from work, and various different medication-related costs. And when we did that, we came up with a number of around 197 million rand to treat these individuals every year. But even this was an underestimate because it didn't take into account people who form part of outpatient programs, people who were not admitted to hospital, people who stayed home, got their own medication. When that was taken into account, the number went up. I'm not sure if you can see this, but close to R1 billion every year, 717 million. Now this again was also an underestimate because it didn't take into account people who have HIV and people who went to private hospitals. And when this was taken into account, the number went up about 3 to 5-fold. So this has become incredibly expensive to the country. And this is the virus that causes the sniffles. Okay, so when it comes to pandemic severity, how do we determine what causes it? And it turns out that there's no one particular factor, that it is unpredictable and dependent on a number of different factors. Things include flu type and subtype circulating, as Ed mentioned a moment ago. Vaccine availability. Is the vaccine actually available in the country? But other than that, how many people are vaccinated? Because it won't help that we have so many million doses of vaccine and only 10% of the population are vaccinated. Then vaccine compatibility. How closely matched is this vaccine to what is circulating? And that can be a big problem. And that is what we find very often, that they are not compatible. And then we have multiple patient-specific factors. We have age, where we find that those most likely infected and those with the worst disease are the very young, under 5 years of age, and the very old, above 65. There are also multiple health-related conditions, which are associated with flu disease severity. We've got immunodeficiency, obesity, asthma, diabetes, and heart disease. And immunodeficiency, obesity, and heart disease are particularly big problems in South Africa. Okay, now when it comes to surveillance, it's conducted by the NICD, the National Institute for Communicable Diseases. Now, they have a mandate to determine what exactly is circulating in the country at any one particular time and report back to the World Health Organization. Okay, and we formed one of 142 national influenza centers around the world. And what the purpose was, as I indicated, was to monitor flu at both an epidemiological as well as genetic level via the Vital Watch surveillance program. Now, this is one of the programs, but there are multiple different programs which all feed into flu surveillance. Okay, so to give you an idea of what Vital Watch does, so what it is that it is a prospective surveillance program that is supported by a network of general practitioners. So these are general practitioners across the country who report of incidences of flu at their practices. And what they do is that they report on it when they suspect it and also provide us with a nasal or oral swab. What we in turn do is that we provide a flu detection and a genetic characterization test. Sorry. Okay, and this is done within a 48-hour turnaround. So what the doctors then get in turn is a free diagnostic test, and they also get CPD points for the HPCSA. And I think that that's probably the major motivation to do this. So the map over here just displays all the general practitioners around the country, and you can see that we've got quite a few in the Western Cape, and all the samples until the end of last year would be sent to our lab at UCT. We'd then process them, provide the answers, and feed all our data back to the labs in Joburg. Okay, so just to give an idea of the stats that we had last year. So in 2018, we received around 1,500 samples from our general practitioners. In the samples, we identified the H1N1, the PDM09. So this is the pandemic flu strain that Ed spoke about. So when you hear anybody creating an alarm that avian or swine flu is still around, please just ignore them. It's nothing big. It is the flu that just hasn't left South Africa. Okay. We also had a number of specimens from the H3N2 type. There were quite a few, and influenza B in circulation. And what we were able to determine is that every year we can determine when the flu season starts, when it ends, when it peaks, what is in circulation, and what is to be taken note of. Are there any viruses suddenly popping up that are resistant to the vaccine? And how will this affect the vaccine the following year? Okay, so what we then do is that all this information feeds into biannual meetings that we have with the World Health Organization. Our meeting occurs in September of every year, where we figure out what is circulating around the world and what is best for the South African vaccine. And then it takes about 6 months to produce the vaccine. Okay, so what we have here is just a histogram demonstrating flu incidences in South Africa. So this is the entire period of last year split up into weeks. And the blue bars are the H1N1, red are flu H3N2, and the green is the influenza B. And what we find is that we could determine that the first week of flu season happened around the end of May. It ended around the end of September. But what was interesting is that we could determine that there were 2 peaks of activity last year. And this is not necessarily the case. So we had a peak of activity peaking in around July last year of flu A, H1N1, and then another one of B, which was around August. And what was interesting is that we could also then split this activity up according to the different provinces. And we found that the influenza B activity was predominantly occurring in the Western Cape. So despite the fact that we're dealing with flu on a national basis, we find that there is some kind of geographic isolation between the different provinces and activities happening in these regions. Okay. So now I'm going to switch track a bit and talk about the different pandemics which hit South Africa. Okay, and the 2 of them is going to be the swine flu pandemic of 2009, and I'm also going to be talking about the avian flu pandemic of 2017, and I'll be explaining why that was important. So as Ed alluded, that this was a genetic reassortment of 4 different flus, right? There were, I'm not going to name them, but there was a North American swine, North American avian, human influenza, and swine influenza typically found in Europe and Asia. And what is important is that this particular virus was not previously circulating in either humans or pigs before it recombined, and was first detected in the USA and Mexico in April of 2009. So for once, China was off the hook. Everybody was looking up there, but it was actually happening over there. Okay, so when it hit internationally, it affected 214 countries. And now I know that my statistic is wrong because there should be 250,000 deaths. So, yeah. So the CDC did get things wrong. Yeah. Sorry, yeah. So no, that initially they said was international. So as I said, it's likely an underestimate, and this is because many did not seek healthcare, and those that did seek healthcare and did die were not actually tested for flu, because very often certain countries do not have the capacity to do the testing. And what we found is that it then hit South Africa on the 14th of June. Okay, so this is where it became interesting. So within the first month, so that's 14th of June to around the 14th of July, there were only 100 cases. But what we find is that 42% of these 100 cases had a history of travel, and they developed symptoms within 7 days of getting back. So that would indicate that it happened slowly because it was all being brought into the country and the virus hadn't established itself yet. So within 2 months, on the other hand, there was an exponential increase in the number of cases. So 2 months later, there were then over 2,000 new cases. Following that, we started seeing 2,000 cases a week by August 2009. And by the 12th of October, we had around close to 12,500 lab-confirmed cases, just showing how quickly this spread. There were deaths in the country. There were around 91, and these were mostly associated with certain conditions, and these were pregnant women and also people with other various different health-associated ailments. Okay. What we did find is that 64% of those symptomatic were under 20 years of age. Now, as we've explained that generally those who are infected are either the really young or really old. And in this case, it happened right in the middle, which is not very often the case. Now, this is an international histogram showing the age distribution for the, oh, sorry, I probably can't see that. So the age distribution for the deaths as well as hospitalizations and basically incidences of illness. I'll just explain what happened. And what happened is that individuals who became sick were mostly above 5 but under 40 years of age. So that is usually the healthy zone. And what people assumed, or how they tried to explain why those that are very old are not getting sick, is that they traced it back to the Russian epidemic of 1977. Now, this was an H1N1 circulating at the time, and they assumed that those individuals may have developed some form of immunity at that time, protecting them. Now, that was an interesting case, and Ed also mentioned this, that this Russian flu was a virus which was essentially identical to another virus which circulated in around, was it the late 50s? And this shouldn't be the case, because any flu virus that is in circulation mutates at a certain rate, developing slowly, adapting to the population. But this virus was essentially identical. So it's assumed that it could have been one of a number of different factors. Either it was a virus that escaped a lab, either deliberately or by mistake, which can happen, or it could have been some kind of live attenuated vaccine which escaped as well. So, but I don't think we will ever really know what it was. Okay, so I'm going to switch track again and talk about avian flu. So what exactly are these? So these are strains of influenza A, or the influenza A adapted to infect birds. The primary natural reservoir are the aquatic migratory birds, so these are very often ducks, and these are known to be subclinically infected. So very often we do not know that they have it, but they are spreading it around the world, and that's a particularly big problem. So there are two different classifications for these. We have those which cause a asymptomatic or mild infection, also known as the LPA, low pathogenicity avian influenza virus. And then we also have those that are highly pathogenic, also known as the high pathogenic avian influenza viruses. And these cause severe disease in birds and result in high death rates. Okay. And these high pathogenicity are known to cause deaths of millions of birds worldwide. So these are either killed by culling or they die naturally. So is there any danger to humans working around infected birds? Now, sporadic infection has been known to occur in the human population with avian influenza, but it is rare. Okay, and a particular avian flu which has been known to infect humans is the H5N1 subtype, which Ed has spoken about. Now, this has specific public relevance in health spheres because up to September 2017, it has infected 860 individuals and killed just over half of these. Now, what is interesting is that this is only associated with people who have had close contact with infected birds, either alive or dead. What is also interesting, or I'll get to that in a moment, and the reason that they picked it up is that this virus is spread in the droppings or the mucus of the birds, which is then aerosolized and can be inhaled or infection by means of contact with any mucus lining, which would then lead to infection. So the interesting point, is that this virus is not well adapted for transmission between humans. In fact, there are very few cases where it's been suspected, but it has never been confirmed. So the only instances are people that have worked with infected birds. Okay, so the virus that spread to South Africa in 2017 is the H5N8 virus, which is a high pathogenicity avian influenza virus. And this virus started out in the south of China around 2010, which then moved out to South Korea in 2014, and these little symbols indicate individual outbreaks that are occurring, either in wild bird populations or in commercially farmed poultry. Subsequent to this, it then moved out to North America, Southeast Asia, and Eastern Europe. And we can then see loads of outbreaks occurring in 2016 and 2017 further west. And then finally it hit Africa in November of 2014. During at the end of 2014 through 2017, the virus then progressively moved further south, where eventually it hit Zimbabwe in Mashonaland in May of 2017. And this is when alarm bells started going up in South Africa because we knew that we might be next. And the big question is, were we ready? No. So as of the 7th of June 2017, 3.8 million birds had either died or had been culled in 14 countries. And this was a huge problem. So when it came to South Africa now, infections were first identified in commercial poultry farms up in Mpumalanga and Gauteng between the 19th and 22nd of June, occurring in both wild bird populations and commercially farmed poultry, but it was first identified in the commercial poultry because they started dying. It was registered as the largest recorded H5N8 outbreak in Africa, spreading to all 9 of the provinces. And between the 19th of June and 31st of October, which is the time span of the commercial outbreak, more than 3 million birds died in 92 localized outbreaks. So these were commercially farmed poultry, wild birds, and also hobby bird farms. And in the Western Cape, we found that 2/3 of the outbreaks was in fact localized here and resulted in wiping out of 75% of the egg-laying capacity in this region, and resulted in a picture that looks something like this, where there were no eggs in Pick and Pay. So what is interesting is that hobby bird farms, like the World of Birds out in Hout Bay, was particularly hard hit. And it turns out that they lost, I think, over 3,000 or close to 5,000 birds during this stint. And I'm pretty sure that they identified that it was because of sacred ibis that landed on the property which were infected. Okay, so mammalian infection. No human infection has been identified to H5N8. So it's classified as a low-risk avian influenza virus for us. Okay, but H5N8 is in fact related to the H5N1 virus, which I mentioned previously. It is evolved directly from it, and H5N1 is known to infect people, so this might pose a problem. Okay, and the increased frequency of H5N8 in birds, and therefore exposure to humans, can increase the probability of a zoonotic event. As you know, viruses simply need the opportunity. They just need exposure, and the evolution of the virus will take care of the rest. So you want to minimize exposure. H5N8 has also been known to be mildly pathogenic and has been shown to infect mammalian models. So they found that they can infect mice as well as ferret models. And one of the bigger problems is that there's absolutely no pre-existing immunity to this particular virus in the human population. So if it does adapt, it will be a particularly big problem. Okay, so because of this, we went out to determine what the zoonotic potential is of this virus. How easily will it transmit to humans working at the interface? So that was the one question. And the next one was, can we see any signs of genetic adaptation of the virus to enable it to infect humans? We usually know what mutations to look out for, which mutations to look out for in virus to determine if it can jump the species barrier. Okay. So this was then a study which was a collaborative effort between the National Institute for Communicable Diseases, the Department of Agriculture, Forestry and Fisheries, the National Department of Health, and the Elsenburg Veterinary Institute. And we worked together to develop a case definition for individuals which we suspected of being infected. We developed a SOP for sampling of people and the testing of samples, and also developed a case investigation form in order to get all the information on people that we suspected of being infected, to try and tie down what associations or disease associations we could find with being infected, if they were in fact infected. And then also we developed information pamphlets for staff working with the birds. And we decided to do surveillance on individuals who were both symptomatic and had a history of exposure to infected birds, satisfying both an epidemiological and clinical criteria. And what was decided on is that only individuals who became sick within 7 days of being exposed to a sick bird were sampled. Okay, and then we also conducted both active and passive surveillance. Active surveillance is where we physically went out to the farms where individuals were showing to be symptomatic and took their samples. Passive surveillance is where we supplied clinics surrounding farms that had infections, and if anybody reported to the clinic, they'd take a sample and send it to us for testing. Okay, so this is a busy table, so I don't want you to really focus on it. The only thing I want to focus on is probably not in your view. So through passive and active surveillance, we identified 74 individuals who turned out to be symptomatic within the time period of being exposed to a sick bird. Okay? 2 of them were found to be infected with flu, but this was not an avian flu. It, in fact, was the seasonal flu, and these 2 individuals died. Okay? So completely unrelated. Now, you might have heard in the media in 2017 that there were 74 individuals identified as being infected with avian flu, and I heard this on the news as well, but it turned out to be a misinterpretation of our data. So we reported on one thing, that we've got 74 individuals who were symptomatic, and they took it as there were 74 people with avian flu, which caused a big problem, quite a bit of panic. Okay, so what we did next is that we couldn't find this virus in the human population. So what we did is that we got a couple of samples of the flu from the veterinary side. So these was from infected birds. And we then sequenced the virus. And what we tried to do is compare this virus to any other virus that has been isolated, or any other H5N8 virus, to see how closely it's related. So don't get freaked out by the next picture. Okay, so what this is a phylogenetic tree. And what it shows you is how closely related different viruses are to one another. When they're lying next to each other in a straight row, it means they are essentially identical. And these over here are all the viruses isolated from the Western Cape. These over here are viruses, H5N8 viruses from around the world. And what we can see is that all our viruses clustered next to one another, suggesting that the outbreak or the pandemic happening in South Africa happened because of the entry of a single type of virus by multiple birds at this one particular time. What we are also able to do is, because it compares different viruses to one another, we can find the most closely related viruses have been identified in India and Egypt in 2016. And the fact that we're able to do this suggests that this may be the same virus that traveled from the north down to us from 2016 onwards. Okay, so just to give a bit of discussion and surveillance, we investigated the risk of transmission of HIV-N8 to humans. 74 individuals were identified by active and passive surveillance, but none of them were found to be infected. And we therefore corroborated global surveillance efforts suggesting that this virus is a low-risk pathogen. The limitation here is that very often we were not allowed on the farms within a particular time period because they were afraid of a possible biohazard risk. So we had to wait until the farmers decided that it was safe enough to enter, by which point individuals who were symptomatic may have cleared the infection. And for this reason, we need to do an antibody follow-up because antibodies are long-lived and will remain in the individual systems for much longer after the virus has been cleared. Okay, and also when we found that the virus is closely related to the Egyptian or Indian viruses, it infers a phylogeographic relatedness, i.e., a genetic geographic relatedness. Okay, when we sequenced the virus, we identified some signs of mutation which would allow it to infect humans. But this does not necessarily suggest that it will infect humans, okay? As we find that you might need a combination of several other mutations as well to allow the virus to effectively mutate or infect humans. And to do this, we are going to be conducting further cell culture-based or animal infection models to determine what the relevance is of these mutations that we identified. So In conclusion, continued surveillance at not only the human but also animal interfaces for the presence of mutations and phenotypes is important for both the control and aversion of potential zoonotic and novel pandemic events. And that's me. Thank you.
Have we any information whether a specific genetic or gene associated with the high pathogenicity of the 1918 flu?
Yes, we do. They've identified that it was in fact a suite of 3 particular genes which stimulated a very strong immune response in those people that were infected. It stimulated what was called essentially a cytokine storm. What you find is that most viruses, when they are killing the individual, it's not the virus's purpose of doing it. It usually happens to be a result of an immune response which just goes completely out of control. And it is that uncontrolled inflammatory immune response which usually kills the people. And that is what happened with the 1918 flu.
And which genes?
I cannot remember it right now.
Hemagglutinin and 2 non-structural.
Yeah, definitely the non-structural ones. Yes.
When you isolate these viruses, presumably they're kept for future reference later. How safe is all this? Because you've spoken of viruses escaping accidentally or otherwise from laboratories. Do you ever sort of get rid of stuff that you've worked on, and how do you do that?
So you'll find that, okay, so this is a twofold answer. So the way in which we treat infectious viruses or anything has changed significantly over the years. You can ask Marie Curie about that. The way that we treat certain things in science has changed. The biohazard containment levels have changed. So at the moment, if we're dealing with anything novel, anything that is potentially a pandemic virus is dealt with in a P3 facility where we control everything. Nothing actually ever gets out. We do store everything. Scientists are the best hoarders. We never get rid of things because we never know when something is going to be worked on in the future or when it's going to be relevant. So we are particularly anal about not letting anything get out nowadays. And if anything does, we are sure to sterilize everything completely. Yeah. Yes. You can never really tell what the outcome of an infection will be, right? There may be a couple of underlying issues which could lead to an exacerbated, disease outcome. But what also happens very often is that one infection could open the door to another infection. And that could be, open up the door to something which led to the other symptoms. So it's highly possible that it could have been because of that, but I think you need to open up the door for more research to figure out what exactly the other underlying issues could have been. Yeah.
My wife and I are known as swallows, and that means that every December we migrate from Sweden to the Cape, and in April we get the flu vaccination here. Okay. After we return to Sweden, we get another vaccination in Sweden. Are we overdoing it?
No.
No, not at all. What you'll find is that there are different flu vaccine recommendations for the different hemispheres. Now, it's not 100% perfect, but because we've got different strains circulating in the different hemispheres, you're doing exactly what you should be. Because what we have down here will not necessarily be what is up in Sweden. So, well done.
So you're not a disease vector, is what we're saying.
Thank you.
What is the future, let's say, for mankind? Let's say our population is growing, people are traveling, which I said, I'm also swallowed. I'm not vaccinated so far. But what is the future? Is this issue of influenza or other diseases getting worse, in your opinion?
I think it's a combination of things. So, firstly, to get into the topic of whether it's worse, I don't necessarily think it's worse. I think it's just being reported more often. I think that our surveillance mechanisms are so up to scratch at the moment that we are more aware of it. But also the future of it, I think that there'd be multiple different things. The one would try and be some kind of vaccine, which is a universal vaccine, so that's something that's not needed to be updated so often. You know, so you'll take it once and it will be good for a few years for multiple different regions around the world. And then the other one is what Ed was talking about is different mechanisms of producing the vaccine. So we've got the plant-based mechanisms and vectors that can produce vaccines at much higher yields than you can produce in eggs. I mean, I've worked with eggs, and they are really tedious to work with. I've also worked with the plants, and you can have a much larger biomass of the vaccine being produced in plants, and I think much easier too.
Ziad had the misfortune of doing a PhD with me 10 odd years ago. And if this very fine book, which you may or may not know, Will actually has got a lot of information about vaccines, but also universal and future vaccines. But that's the point, is that the vaccines we make right now are very specific. The course of vaccine research right now is to go less specific, something that will protect against everything that's circulating and for the foreseeable future. And it is possible to do so. It's actually been shown in animal models that some of these things do actually work quite well.
Just a comment rather than a question. Professor Becker, all those years ago, was walking on Musenberg Beach, and he happened to be a very observant gentleman. He noticed that there were a lot of dead, small, white birds on the beach at Musenberg when he went for a walk on the beach. He picked one up with his bare hands, and he took it into the laboratory where he's working at the University of Cape Town, and from that he isolated Hearn virus, that very early one that Ed showed us here, that was picked up on Musenberg Beach by an observant person. The common antigen, is there a group common antigen that could be used to make a universal vaccine? Yes. And if so, why has it not been used?
Is there a group common antigen that you could use? The answer is yes. You all, when I showed you pictures of the virus earlier, there's a minor protein called M2. There's another one called the matrix protein, or M1. Both of these are far more group specific than the hemagglutinin and the neuraminidase. Problem is they're a lot more complicated to make by conventional techniques. However, if you go to the new techniques, these are in fact incorporated. And there's a Canadian company called Medicago, which actually uses it in the production of their plant-made vaccine, because they make what look like virus particles, but in fact come out of a plant and are not infectious. They use